Hello everyone. Today we are going to learn class 9 biology. Chapter 5 Fundamental Unit of Life Part 5. In this video we will learn transport system of the cell, that how the molecules transport, that is diffusion. As in our previous lectures, we had learned that plasma membrane allows the material to enter and exit outside of the cell. But how the materials get entry inside the cell, and same way how the materials come outside of the cell. This whole process is done by transport system of the cell. There are two types of transport system: active transport and passive transport, diffusion and osmosis. In this video, we will learn about the process diffusion. The diffusion is a movement of molecules from a region of higher concentration to a region of lower concentration. It occurs in solids, liquids, and gases. Let's look at the example of diffusion in liquids. What happens if we put a drop of ink in a glass of water? Single dark color ink drop spreads when added to water, and intense blue made slightly. Means the whole glass turns into intense blue color. Actually, when we add a drop of ink at a particular area, ink molecule concentration is higher at their area. You can see in image, these molecules move towards the surrounded area where only water molecules are present and not the ink molecules. Hence, all water turns into blue color as ink molecules moves from higher region to lower region. Now, take example of diffusion in gases. That how gases transport across the plasma membrane. Let's take example of carbon dioxide. When cells are doing cellular respiration, lots of carbon dioxide accumulates inside the cell. On the other hand, there is very little carbon dioxide present outside the cell. Hence, carbon dioxide molecules moves outside the cell. Now, there is very little oxygen left inside the cell after cellular respiration. So, the oxygen from outside the cell moves into the cell through plasma membrane. It means carbon dioxide moves out and oxygen moves in. Both the gases moves from their area of higher concentration to the area of lower concentration. And this process of diffusion does not require any energy. Diffusion is a process by which transportation of many substances carried out by plasma membrane. Diffusion is not only the process of transportation. There are other systems also. We will learn other process in next video. Now look at some day-to-day examples. If someone is cooking something delicious, you will get that smell of it from meters away, which means that smell spreads through the air. We burn such kind of mosquito coils at our home. At first, you don't get the smell of burning unless you stand so close to it, but gradually it spreads throughout the room. After a few minutes, you will get that smell anywhere in the room. It means that smoke spreads in all directions. Because of this diffusion, smell, gas, smoke, etc. spread through air easily. Because of this process, if you add color in water, it will spread throughout equally. Because of diffusion, flavors of tea spreads throughout the whole water in a cup when you put a tea bag into a cup of hot water. Now we have to learn the second cell transport system, that is osmosis. This process also helps the material to enter and exit from the cell. But before osmosis, we have to learn about solution. So, we'll see what is solution. It is solute plus solvent. Now, what is solute? It is the minor component in a solution that dissolved in the solvent. And what is solvent? It is a liquid in which a solute is dissolves to form a solution. Let's take an activity to understand solute, solvent, and a solution. You all have made lemonade once in your life. So, for making lemonade, first we have to take one glass. We have to fill the glass with water. Squeeze lemon juice into it. So here, look, you can see that lemon juice is dissolved in water. Means we cannot distinguish lemon juice and water separately. So, lemon juice is known as solute, and as water is dissolved lemon juice in it, it is known as solvent. Further, we are adding sugar crystals into it and pinch of salt. After stirring it sometimes, we can see that sugar crystals and salt dissolve into water. It means that sugar and salt are the solutes as they are dissolved into water, and water is called as solvent. I hope you are clear with the solute, solvent, and solution. And this whole is called, the lemonade is called as solution. Now we'll move up to osmosis. What is osmosis? It is the movement of water through a semi permeable membrane from a region of high concentration to a region of low concentration. It means water molecules go in and out of the cell by this process. To understand properly, let's take an activity. Do you know raisins? It is also called as kismis. What happens if we put this kismis into water for the whole night? If we soak these raisins for the whole night, then next day you will observe that dry raisins now become swollen raisins. How this magic occurs? It is not the magic, it is done by the process osmosis. Actually, what happened when we put dry raisins into water bowl? Dry raisins have less amount of water. So from surrounding, water enters into the cell of raisin to helping them to swell. And this is done by the osmosis that the water moves from higher water concentration to lower water concentration through a semi-permeable membrane. Now we have to perform an activity of the action of different type of salt solution on living cell. So here in three dishes we had kept red blood cells. You can see in first plate the red blood cells are shrinks, whereas in second there is no change in the shape, and in third the cell gets swollen. Why this happened? Let's see. In the first case, the water content in the surrounding medium is less as compared to the cell placed in it. In other words, content of salt is more in solution and hence it is called concentrated solution. If the medium has lower concentration of water than the cell loses water by osmosis and shrinks. Such solution is known as hypertonic solution. That is the solution with higher salt concentration. Now look at the second case. Here, the content of salt and water is same in either side of the cell. Means it is balanced. So here what happened if the medium has same water concentration as the cell, there is no net movement of water through the membrane and cell stays at the same size. Do you know what such solution is called as? Such solution is known as isotonic solution. That is, the concentration of water is same on either sides of the semi-permeable membrane. Now look at the third set. Here the cells are surrounded by dilute solution, means less content of salt and more amount of water. If the medium surrounded by a cell has higher water concentration than the cell gets water through osmosis and swell. Such solution is called as hypotonic solution. That is the solution with higher water concentration. Hypo is a Latin word which means below. Iso is a Latin word which means equal. Hyper is a Latin word which means more or above. You can see clearly in this image. In hypotonic solution, the cell swells and bursts. There is no change in isotonic solution, and in hypertonic solution, the cell shrinks as it loses the water outside. Now we have to learn plasmolysis. It is a process of osmosis which occurs in plant cell. It is defined as the process of contraction or shrinkage of the protoplasm of a plant cell when it kept in hypotonic solution. Protoplasm is a liquid fluid part of the cell which we had learned previously. Plasmolysis was generally derived from a Latin word plasma means the mold and lysis means loosening. As plant cells have the cell wall, we can easily see the shrinkage of the protoplasm in the plant cell in a spherical shape. You can see in this image that how normal cell shrinks after kept into the hypertonic solution. Let's perform an activity to understand plasmolysis properly. Plasmolysis is a shrinkage of fluid part of a part cell as a result of loss of water of the cell through osmosis. We can see plasmolysis in laboratory by using salt solution or sugar solution. Normally, for this activity, royo plants' epidermal cells are preferred as colored cells clearly seen under the microscope. Materials required: royo leaf, forceps, glass slides, cover slip, compound microscope, needle, droppers, sodium chloride 0.1%, sodium chloride 5%. Procedure: take two glass slides and place it on the table. Take a rayo leaf from petri dish, fold the leaf and tear it along lower side of the leaf with the forceps. Pull out a thin, transparent segments and put on the glass slide. Using a dropper, add 0.1% sodium chloride on one glass slide. Using another dropper, add 5% sodium chloride on the second glass slide. Place the cover slip over peel of leaf using needle. Place the slides under the microscope and observe it. Observation: after half an hour, we can see that cell that kept in hypotonic solution appear turgid, whereas the cells kept in 5% sodium chloride solution show plasmolysis. Conclusion: when plant cell immersed in a 5% sodium chloride solution, water moves through the cell because water concentration inside the cell is higher, whereas outside water concentration is low. Ultimately, Protoplasm causes shrinkage and assumed a spherical shape. This is the real slide of the shrinkage of the protoplasm of a plant cell.